The first thing I should say is that the real users here are Harry Thorpe and Sean Bayliss. Well, the story started about a year ago. I invited myself up to Birmingham, bought Nick a coffee, and in a weak moment he gave me a he registered me on the system. Nothing happened for a couple of months, and then I happened to mention it to Harry and Sean, and they got going with it. And it has, it's no exaggeration to say, it's absolutely transformed what we can do. We can work on data sets which we just wouldn't dream of entertaining the thought at all of working with before we had access to this system. So it really has been fantastic. We haven't installed anything particularly exotic. We haven't we've just used the sort of standard tools that you use for mapping and assembly and annotation and all the rest of it. We haven't. I know these things will come pre-packaged on GVL and that other thing that was mentioned, that Tom mentioned, where you just press a button and it's all done for you. But Sean and Harry have basically built this up by installing these things in a sort of more bespoke fashion, and it's worked absolutely fine. So I asked Harry and Sean for just some comments before I came here, and that I could relate to you. Both, of course, were, as I say, were completely positive about it. Harry saying it's an amazing resource. In particular, the fact that you have the VMs makes it so much more convenient than the normal sort of queuing system where you have to sort of wait for some time on the machine. It's basically like having, of course, your own local computer, essentially. There are some uncertainties, which I gather, I wasn't here yesterday, so I gather that the strategy has been thought out about how these users are going to be managed in terms of VM assignment and what happens if they're just sort of left open without doing anything, that sort of idle time. So in that sense, potentially, it could be a bit inefficient if those VMs are sort of open and not doing anything, but that's for you guys to sort out. We don't mind that. And we were quite sort of flattered to be tagged as power users on the system. I thought that was quite cool. So there was, Sean made the very good point as well. He recently helped out with a bioinformatics workshop in Finland and found it somewhat frustrating that there was a very different background amongst the attendees there about just use of Linux and the operating system, and just having to get people up to a certain standard before you could do anything. So for workshops, for Climb is just fantastic because everything's standardized and you can get things going, have everyone on a level playing field before you even start. So that was pointed out as a very big advantage of the system. So essentially, as I say, it's completely changed the way that we can operate in our setup, and I'm sure that will be the same for many other users as the system grows. So that's my user's perspective, and now I'm just going to go to more or less a standard research talk. But this is a project which has used Climb as an integral part of it. There's been a couple of other projects where we've used very big data sets, but I thought I'd keep it simple and just talk about one project here today, but you can ask me about others that we're doing later on the day if you like. So essentially this project asks the question. This is Harry's work that he's doing for his PhD, and the question is, how much should we care about intergenic sites? So it's struck me for quite a long time that there's, in the genomics era, since the dawn of the genomics era, and before possibly, there's been something of a blind spot when it comes to intergenic regions. They are about 10 to 15% of the genome, quite a surprisingly consistent proportion actually in different species. Of course, they're non-protein coding, but they're not non-coding in the fact that they don't do anything. But at the moment, even though there are many, many examples, and I'll just mention a couple in a minute, in the literature of where regulatory elements in intergenic regions have been shown to be extremely important in terms of function, in terms of antibiotic resistance, in terms of virulence, there's no these are always considered as kind of isolated exceptions, almost. There's no broad quantitative understanding of how much intergenic regions really matter in terms of function. And I think this has been due to a number of reasons behind this. There's certainly a legacy from MLST and the very sort of gene-centric view of the world, which has kind of led on to the core genome MLST type approach in this gene-by-gene system, which seems to be now becoming pretty much established as the way that we should manage these big genomic data sets. And really what that means is you're just cutting out all the intergenic regions. And I can see why that this is kind of gone this way. But I think there's a real danger that we're actually, this 10 to 15%, we're actually going to miss quite a lot of important, if we're interested about drawing genotype-phenotype links, then we're actually going to be throwing away quite a lot of very important information, potentially. I think there's also a perception that intergenic regions are somehow much more difficult to work with, that they're messier, that the alignments aren't so easy to make because there's gaps, etc. And that's true to an extent, but I think that it's probably not as messy within an intra-species level as you would imagine if you actually go and get your hands dirty. You can quite easily define a sort of core set of intergenic regions which align pretty well, which we've done for this work. There's also, of course, when you're talking about selection, what's the way we routinely measure selection on coding regions? The most common method is, of course, to calculate DNDS, which is the ratio of non-synonymous to synonymous changes. If that ratio is very low, we say that gene is under selective constraint. Synonymous changes are much more common than non-synonymous changes. But of course, we can't do that with intergenic regions because it doesn't make sense. There are no such things as non-synonymous and synonymous changes. So there's a perceived lack of methodology whereby it's thought that it's actually should be quite hard and a lot of effort to think about measuring selection on intergenic regions. But in fact, as I'll show you, you can use exactly the same logic for intergenic regions, and in fact, there's no real problem at all with doing that. So just a couple of examples. This is one more or less picked at random from the literature in quite an important one in Mycobacterium and tuberculosis. There's lots of homoplasic mutations identified in the promoter of this of the EIS, which are responsible for kanamycin resistance. There's another GWAS study that was carried out by Ruth Massey in Bath on a particular clone of MRSA, ST239, looking at, as Sam has just described, looking at associations with laboratory phenotypes, in this case toxicity, as measured in the lab. And Ruth found 13 intergenic SNPs, which were unknown before, but significantly associated with different changes in toxicity. And she went on and experimentally confirmed 4 of these by transposon mutagenesis. So when you're looking at GWAS, you know, the intergenic sites come up regularly. I mean, it's not a big surprise to see them coming out in these sort of studies, which all points to the fact that actually we should really be paying more attention to intergenic regions than we are. There's not only promoters and terminators, there's also small RNAs, which act either as a sort of antisense RNAs binding to target transcripts or interacting directly with proteins. And there's a whole body of literature, I'm sure many of you are very familiar with it, that they have all sorts of diverse roles in biofilm formation, virulence, plasmid replication, transposition, all sorts of different responses to different environmental signals. There's different aspects of metabolism, protein secretion, and so on and so forth. So small RNAs really seem to be, in a sense, the cryptic dark matter in terms of functionality embedded within these intergenic regions. So when Harry started his PhD, this was his brief, to develop, hopefully, rapid and easy and useful, meaningful methodology to try and quantify how much purifying selection is operating on intergenic DNA. We're using synonymous sites as a sort of baseline throughout all this work. We're assuming, so everything is compared to what's happening on synonymous regions encoding in genes. Doesn't necessarily mean that synonymous sites themselves are neutral. In fact, we know they're not. There's codon biases and all the rest of it, but they're the most convenient proxy for neutrality that we have. So, in fact, our estimates of constraint, this makes them conservative because we're not taking into account the fact that synonymous sites themselves are under selection. So we wanted to compare lots of different populations that can contrast markedly in terms of population structure and rates of recombination. Our strategy for controlling for any of the potential confounding effects of recombination and clonality and all the rest of it was just to look at lots of different species that have a wide range of recombination rates. And we also wanted to see if we took intergenic regions and we took out every possible site that we could assign any sort of function to, promoter or terminator, a small RNA, using various prediction programs, and just look at what's left, then is there still any evidence of selection operating on these regions? So these are we looked at 6 species. We started off looking at 2, and then we moved to 6, and we realized that once all the scripts and everything were in place, it's actually not such a big job just to expand the analysis, and Harry's done a great job in putting all the scripts he's used available on GitHub, and when the paper's finally accepted, it's under consideration at the moment. All their work will be available for anyone else to basically, hopefully, repeat this analysis. So the species that we considered were E. coli. This is where the data came from. So for E. coli, we just looked at all the complete genome sequences. There's 157 in NCBI, which are just all nicely closed and complete, so that was easy. Salmonella enterica, we used the Public Health England data. We were given permission to actually use CLIME to map and variant call 8,000 of the Salmonella enterica data produced by the Public Health England, which took about a week to do on CLIME. We couldn't do that just on our laptop very easily. From those 8,000 or so, we picked out 366 representative ones, so we're not sort of using lots of genomes which are practically identical. We used the Holt et al. Data for Klebsiella pneumoniae, so that's quite a diverse collection, which was published in PNAS last year. Staph aureus data came from a big study that I was part of, looking at MRSA over in the UK. So there's the whole data set, again, is about 3,000 genomes. We just used took representative examples from each clonal complex, 133 genomes from there. The pneumococcal data came from the Mailer study, from the Sanger, Chua Preacher et al. Again, we took 264 representative ones of those, and the TB data came from the Kaseli et al. Data, which was the Russian drug resistance study. Again, the whole data set's, I think, about 2,000 genomes, and we picked 144 representative ones. So we found, Harry, for each of these data sets, identified a set of core intergenic regions, as well as a set of core genes, taking into account a threshold, below a threshold level of ambiguous characters, and their distribution had to be in at least 95% of all the isolates to be counted as core. So the first thing we did was look at the sequence characteristics of the genes and the intergenes, and of course the genes are bigger. This is just size. There's 3 types of intergenic regions according to the orientation of the flanking genes. So here we have coordinated gene orientation, so the genes going the same direction either side of the intergenic regions, and these are quite tight size distribution at about 150 bases or so. genes which have a double promoter, so the genes are going out this way, tend to be, these intergenic regions tend to be a bit bigger for whatever reason, and then double terminators, there's more scatter there, but generally they're the same size as the co-oriented ones. The size of genes themselves, if anyone can tell me why genes should all be the same size in these different species, a very tight distribution of just that. Of course, we all know this, but can anyone actually tell me why genes should be just under a kilobase big? If that's something fundamental about information theory, I'd like to know. But nevertheless, that's true. So there's also a difference in GC content between the genes and the intergenic regions. It's all been described before. Intergenic regions are slightly lower GC, except in TB, which it drops very, very, very slightly, but really not very much at all. And TB is, of course, as I'll be talking about, an exceptional species for lots of reasons. So the first approach we use is a really, really simple idea. So the idea is that, I guess many of you will have noticed this, if you have a large genome data set, maybe 100, 200, 300 isolates, and you build a tree, and you'll notice that many of the SNPs are rare. They're only a sizable proportion, maybe over 50% of them, you only see in one of your genomes, okay? So this is based on, if anyone's familiar with Tajima's D and site frequency spectra, this is based on a very established method that you can actually use the rarity of mutations to tell you something about selection. And the idea is very simple, that if you have a mutation which isn't so bad for the cell that it kills it immediately, but it's bad enough that it won't actually rise in frequency in the population, then those sort of mutations are going to be more rare than you would expect under strict neutrality. So the more, if you have an excess of rarity, then that's good evidence that you actually have ongoing purifying selection, okay? So you can actually say how much selection is going on in a particular class of site just by asking how many of the mutations you see at that type of site are really rare. In other words, are only found in one of your genomes, or a singletons. Okay? So we did this for the different classes of mutations. We looked at our nonsense mutations, which is expected to be under very strong selective constraint, therefore most commonly rare, if you can say that, non-synonymous, synonymous, and intergenic. So when you do this, you see for 5 of the 6 species, you see a very, very consistent trend. So the purple bars, these are the different species: E. coli, Salmonella, Klebsiella, Staph, Strep, Mycobacterium tuberculosis. The purple bars are nonsense mutations. So what this is telling us is that about 50% of all the, is that 50? Does that say 50? 80, sorry, beg your pardon, 80% of all the SNPs that cause a nonsense mutation, you only see once in the data set, okay? The next category where you have the most rare proportion of SNPs, the highest proportion of SNPs that are rare, I should say, are non-synonymous, which you'd expect to be under selective constraint, of course, and then intergenic SNPs very clearly become intermediate between non-synonymous and synonymous in all these 5 species. So we can say immediately with this very, very simple analysis that the amount of selective constraint on intergenic regions falls pretty much in between, intermediate between the amount of selective constraint on synonymous sites and the amount of selective constraint on non-synonymous sites. So tuberculosis doesn't really work. There's no sort of signal of any sort of selective constraint on anything, frankly, or at least there's no difference between the sites. So it didn't really pan out for TB. The other method we use, so I've already mentioned DNDS as the classic way of measuring selection on encoding regions. So all you do is measure, instead of measuring DNDS, you measure DIDS. DI being the number of SNPs, intergenic SNPs per intergenic site. So exactly the equivalent, except you have to do it on a genome-wide level. So you compare the whole DI over the whole genome with the whole DS over the whole genome. Because you can't compare single genes, obviously. So we did this for all the 6 species, and we also looked to look for the effect that we've described about 10 years ago, and I've been banging on about ever since, where you can actually plot the action of purifying selection over time when you're thinking about DNDS. What happens is that when you have very, very closely related genomes, you tend to have, which maybe differ at like 10 SNPs, the proportion of non-synonymous SNPs in that comparison tends to be much higher than when you have a much more highly diverged comparison. And the reason is, as I was saying, that you have the effect of purified selection is an ongoing one over time. So de novo mutation doesn't care if it's producing a non-synonymous or a synonymous site, okay? So when you're seeing very, very recent mutations, you see much closer to what's actually the mutational footprint before selection has had a chance to change this ratio. So you see a much, proportionately a much higher number of non-synonymous changes, and then the selective sieve operates over time to preferentially remove the non-synonymous sites, so the ratio drops over time. And this is something that you can observe in lots of different populations. This is a simulation. The slope of the curve is dependent on S, how the selective coefficient, how bad the non-synonymous changes are, and the effective population size. And all this is established 40 years ago, the nearly neutral theory by Ota, 71. It's all exactly what she was talking about, except now we can plot it beautifully. And you see this is real data in this original paper back in 2006, where you see different trajectories. You see a much steeper trajectory for E. coli than for other species, and we made up a story at the time that this was probably because the population size of E. coli is very, very big, so that this sieve is acting a lot more efficiently, because as you all know, selection is a lot more efficient in big populations than in small populations. So we did this for a couple of our species. Instead of the red line is DNDS against DS, so it's divergence time, and the blue dots are DIDS against DS. So you can see in both Staph aureus and pneumonia, the slopes are going down. DIDS is higher than DNDS, so there's not so much, the selective constraint isn't as strong on intergenic sites as it is on non-synonymous sites, as we just shown in the previous analysis, but nevertheless, this ratio is always under 1. So there's much more selection on intergenic sites than there is on synonymous sites. We took another approach where we just said, okay, let's not worry about the continuous time plotting against DS. Let's just look at essentially two time points within clonal complexes and between clonal complexes. So the red boxes here represent very, very short time frames where both DIDS and DNDS are higher than the equivalent comparisons over longer time frames, which are the blue comparisons. And this is a consistent trend over all the species, showing that you do, again, you have this ongoing purification of intergenic mutations over time, just as you do with non-synonymous mutations. So, for any given value of DS, by and large, DIDS is greater than DNDS, so the selection is there, but it's not as strong. DIDS is less than 1, so there's more selection on intergenic sites than on synonymous sites, but not as much selection on intergenic sites as non-synonymous sites. Okay? Good. So, once you've got these parameters, DI and DS, you can actually very simply calculate the proportion of all the sites, both non-synonymous sites and intergenic sites, that you predict to be under selective constraint. Okay? So it's very simple. You just find the difference between DS and DI divided by as a proportion of all DS and then multiply by the number, total number of intergenic sites. So what this is saying is for all these species, somewhere for E. coli, 94.6% of non-synonymous sites are under some level of selective constraint that we can detect a signal for. For E. coli, 78.7% of intergenic sites are under some level of selective constraint. So E. coli seems to be showing a stronger footprint of selection than the other species, okay? Just as it did in the original DNDS study, I should say. So that kind of fits, possibly because it's got a high population size. For Staph aureus, which is showing the weakest level of selection, apart from TB, which I'll come to, we still have, so about 90% of non-synonymous SNPs are under constraint, and still 61% of all intergenic sites are under selective constraint. Okay, we can just plot these as a nice, slightly more visually appealing bar chart, just showing the proportions. The dark red, these are non-synonymous sites, the red and the pinky one. The dark red is the ones under constraint, and this is not under constraint, and it's the same for the intergenic sites at the top. Next, we went and had a look and said, okay, so roughly about 70%, on average, 70% of all intergenic sites are under selective constraint. Let's go in and see if that changes when we look specifically at all the different regulatory elements that we can identify. So we use all the annotation we could get hold of, all the prediction programs that we can use. Harry put a lot of effort into making this as meaningful and accurate as we possibly could. A lot of the promoters and the terminators are predicted, I should say, so there is a caveat here. But nevertheless, these are the results. So if we look at non-coding RNAs in all these species, then the proportion that are under constraint is basically the same as the proportion that are under constraint for non-synonymous sites. So we're looking at upwards of about 90%. The next category in terms of the strength of selective constraint are promoters, and then terminators. Terminators don't seem to be, weirdly enough, under much more selective constraint than all the sites that are left over when we take all these non-coding RNAs, promoters, and terminators out. But what's amazing, really, is this column. So we've taken everything out that we can think of, all the promoters, all the terminators, all the non-coding RNAs, and we still, in E. coli, are suggesting that 78.3% of intergenic regions are under some sort of selective constraint. So there's a massive, massive amount of cryptic functionality in these regions, which we just have no idea about. Maybe there certainly are a lot of small RNAs that haven't been identified, because they're being described all the time. But it's almost certainly, even if these are slightly over-exaggerated, and there's no reason why they should be that we can think of, but it's still, I'm absolutely confident that the majority of sites that have no known annotation, no known function in intergenic regions are actually under selective constraint. This is just the same data again, replotted as nice plasticine bar graphs. The non-synonymous, this is the proportion of sites under selective constraint, so non-synonymous, non-coding RNAs. Apart from these two species, Salmonella and Pneumococcus, the other three species, again, as I said, the level of constraint in non-coding RNAs is pretty similar to non-synonymous. Then come the promoters, then the terminators, and then the unannotated sites. So what I didn't show you is the actual data for the DIDS for all the different regulatory elements, and this is the graph here. It's much easier to show it in terms of numbers and broken down as estimated proportions of constrained sites, but here is the actual figures for DIDS for the different regions, the different types of element for the different species. So I would draw your attention to tuberculosis, which I haven't mentioned for the last few slides. I've actually missed them out of the table. I was waiting because none of what I've said about selective constraint seems to apply to tuberculosis. But when we did this analysis, looking at all the non-coding RNAs, all the promoters, the terminators, and all the unannotated sites in the TB data, and calculated DIDS, something surprising happened, which was that we found very strong evidence for positive selection in promoters of TB. So this is almost 2, this value. So TB, as you know, is an extremely monomorphic species. There's hardly any SNPs in our data set. There's not enough to make the between clonal complex comparisons, for example. So how does it adapt? How does it change function? How does it evolve, essentially? So we would posit that actually a major mechanism by which this bug adapts, at least over the short term, is by changing promoter regions, by positively selecting changes in promoters. And this is extremely highly statistically significant. This is just a distribution of DIDS over different pairwise comparisons. This is one here. We expect no selection, and I think something like 80% of all the comparisons, maybe more, are higher than one. And we've gone in and have a look at where these SNPs are in promoters, what the downstream genes are, and it's just your ideal wish list of transcriptional regulators and resistance and virulence genes that you would hope to see. So that we're sort of keeping our powder dry a little bit for the follow-up paper to explore this in a bit more detail with more experimentally validated promoter regions and look at a lot more data as well. So just to conclude, so we can measure selection on intergenic sites. It's a very useful thing to do. You should take more notice of intergenic sites, and if you take everything out that you know about, then you still have an average of 69% of all intergenic sites seem to be under some sort of selective constraint. In those 5 species and in TB, where there's no real evidence of selective constraint, there is evidence of, very strong evidence of positive selection operating on promoters. And that's that. Thank you.
All right, thanks, Ed.